everyone. Welcome to our monthly Science of All Proteins seminar. My name is Bianca Dada, and I'm the Senior Scientific Partnerships Manager at the Good Food Institute. For those of you who are here for the first time, welcome to our community. The Good Food Institute is a nonprofit think tank 100% driven by philanthropy, and our goal is to build a world where alternative proteins are no longer alternative. If you're looking to take a deeper dive into the science of plant-based fermentation or cultivated meat technologies, check out our open access resources at gfi.org/science. With that, I'd like to introduce our first speaker for today. We'll start with Dr. Valerio Hoyos-Villegas, who leads the dry bean breeding and genetics program at Michigan State University. His program develops high-performance dry bean cultivars that are resilient to environmental pressures and offer strong processing characteristics and nutritional quality, including high protein content, both for industry and consumers. His group conducts research in dry bean genetics, genomics, and statistical and population genetics, as well as high-throughput phenotyping for agronomic traits and processing quality. His goal as a scientist is always to generate novel applications and construct skills to improve processes involved in the plant breeding cycle while generating new knowledge that addresses questions that take the scientific community closer to the understanding of plant genetics phenomena across species. Thank you so much for being with us here today, Valerio. I'll turn the floor over to you.
Thank you very much, Bianca, Renee, and GFI for giving us the opportunity to speak today and to share some of our work in dry beans in this world of dry beans and uh, its service to the U.S. Economy, society, and world and globally as well. So, right. So my name is Valerio Hoyos-Villegas. I'm an assistant professor in the Department of Plant, Soil, and Microbial Sciences at Michigan State. Now, I am the PI leader for the dry bean breeding and genetics laboratory. Today, I'd like to sort of give a bit of a mix of flavors of some of the important aspects that take place in our work, both from the applied side, which is the breeding program per se, as well as the some of the research that's ongoing in the lab to give you some idea of how we utilize science and technology to tackle some of the problems that face us in solving some of these global challenges as it pertains particularly to beans. So first of all, I think it's quite this obvious, or I hope it's obvious to this audience, that our effort, our ability to keep up with food production relative to demand and population growth around the world is insufficient. And some of the most comprehensive studies have indicated that current production is not meeting demand at about a rate of about 1 to 1.6% per year gap. So that means that the on-farm production and whatever technology is being deployed into those systems is not keeping up with what is expected to be demand due to population growth at a linear pace by 2050. And so this poses several questions and challenges, of course, but before I move into those, first um, I'd like to first talk a little bit about the importance of dry beans in the U.S. And some of their particular challenges and how that relates to this more broader challenge. So dry beans are a major specialty crop in the U.S. The U.S. grows up to 10 market classes that we can see here in the bottom: pinto, black, navies, dark reds, and so forth. And it produces in the order of 9.5 million hundredweight for example, some of these classes like pintos, which are one of the most produced. However, you can see that there is great variability in the recorded yields for many of these classes. So, for example, pinto is among the most produced but lowest yielding, and black beans are the second most produced in the non-irrigated areas, but it exhibits some of the highest yields in these areas. Okay. So this can raise questions around resource use and um, breeding for those conditions where, for example, irrigation is not available, such as eastern, so our areas here in Michigan. Michigan, in fact, leads in black and navy bean production as of 2025. It swaps back and forth between the second and third largest producer. It's traditionally been the second largest producer overall of beans in the United States and the top producer of black and navy beans. But another particularity about Michigan bean production is that it is also quite diverse in the number of market classes that it grows and markets into the U.S. and global markets. So up to nine market classes are grown around the state. So I mentioned so of these we have blacks, navies, and then a third class would be small reds, and then a number of other sort of smaller classes. Most of our production is concentrated in what we refer to as the thumb of Michigan, is that sort of eastern portion of the state. 80% of the production is concentrated there. Most of these are black and navy beans, but there some of these other market classes are typically found in the middle of the state. Um, Around the center here, just north of where, about 2 hours north of our campus here. So producers in the state of Michigan have at their disposal both an industry that enables them to market beans, not only that, but also extension agronomists that help them produce a what is widely considered around the world as a premium brand, and train and this ability to train and transfer knowledge to our growers so that they have the right amount of attention to detail to produce a premium brand has set the Michigan growers apart worldwide. Not only that, but also they have access to shipping routes through the Great Lakes system, the nautical system, as well as policymakers here in the state capital of Lansing and researchers such as ourselves here at Michigan State University. The Michigan bean crop value is around 250 million presently. The U.S. Bean crop value is around 1.1 billion. One thing that you might I want to point out here is how the getting more into the scientific matter of the talk is how Michigan has managed to essentially not only stay on par with the national average, but also surpass it for several years going now. So Michigan, and our program here has made tremendous gains in producing technology that enables growers to produce beans above the national average. Um, and, and I'll talk about essentially how this happen. The Michigan State University bean breeding program dates back to the late 1800s. So now we could say that we've been running for over 100 years. And essentially, this program has been handed over from multiple faculty members here at Michigan State across several essentially careers. And each one of those breeders have kind of made their mark by incorporating major pieces of technology into the varieties that have been deployed to growers in Michigan and the United States. So I have here some of the milestones I highlighted. And here in the 90s, I'd just like to focus on one, this question of plant architecture. So we've been a driver of germplasm technology to maintain the competitiveness of dry beans, and this has been a major reason why beans have been maintained above average yields. So now this question of the upright architecture phenomenon, let's say, in beans, we in the germplasm and genetic variation that exists within the species, we frequently identify at least more here in temperate production, we identify, let's say, three growth habits. We have type 1, which is a determinate plant, type 2, which is what we call an upright indeterminate or semi-indeterminate plant, and then a type 3 prostate indeterminate. So these first two, they grow up like you would like you if you would imagine, say a soybean crop or something like that, where the plant grows up and plants sort of support each other as well, particularly in this type 2 upright semi-indeterminate type plant. Prior to the 80s, most production in the US, particularly in seeded or medium seed sizes such as pintos and other medium sized beans, were mostly type 3 prostrate, so they would grow along the ground. And this meant that if you're a farmer growing indeterminate prostate beans, you had to, in order to harvest them and market your beans, you had to conduct several operations. The first one was to pull the beans with a machine at the time older than this, but they still exist, which is called a bean puller, which essentially pulls the beans off the ground and then turns them over, where they would then be windrowed by a standard sort of forage hay rake, and then they would be left in the field, not to dry, but also to in full exposure of the elements. So if it rained, these windrows would also get wet, and also they would be in contact with the ground, causing them to spoil, rot. And if you manage to get the right conditions for beans to dry, then you could go in with your standard combine and combine them, and just like you would any other crop. You would combine these windrows, and then you'd get your crop. When breeders at this in this program managed to move some of this behavior, some of this phenotype from the determinate or semi-determinate upright varieties into, say, these pinto beans that were prostrate, bean growers now only had to basically conduct a single operation of simply just combining them like they would any other of their crops, such as wheat and soybeans. And so this meant less traffic over the same field, reducing compaction as an indirect effect, and now if we were to measure that, potentially reducing emissions as well. But not only that, it meant also that the yield losses were far smaller, and also the due to the ability of the plant to be bred to support a greater amount of yield, but also the post-harvest losses were lower as well upon cleaning the beans and so forth. So this was one of these game-changing sort of technologies that came about that has enabled the industry here to maintain competitiveness. So the Beam program has released many varieties over the years. These are just some of our recent releases. Here's our website, and you can see all of our bulletins showing off the attributes of some of the varieties and what they possess and what they don't possess as well. And so this is just a way of us communicating to growers what new technology is available. And so this is another arm that we have at our disposal, is the ability to extend technology and knowledge over to growers through the MSU extension system. Now, I'm switching gears a little bit. Beans, in relation to other crops, have maintained around 1% growth or rate of genetic gain or increases in yield, let's say, um, over time, over long periods of time. And these are based on experiments that are where a group of researchers get together and they grow side-by-side trials of old versus new varieties, and they just simply estimate the rate of growth. This is by no means an estimate of the genetic properties of varieties and whether their genetics um, are the main cause, but it's an indication. Okay. In other crops, there's been far greater growth than in, for example, in beans. But one thing to keep in mind, for example, is that in this country, for example, the land utilized for soybeans is some of the best land available, and a lot of the time, dry beans don't are not cultivated in these areas, and therefore we make gains despite that. So as we compare this relative to some of the demand that I posed earlier, we can see that that scientific evidence suggests also that the rate of growth maintains a gap with the demand. So how do we further increase rates of yield gain in beans? And this is essentially my job here. How do we keep pushing this up further? How do we come up with new ideas? And I'd like to illustrate that by simply showing this framework that is commonly used in our discipline, which is called it's the breakdown of phenotype. So phenotype is made up of a genotype. So it's basically the individual genes or the genetic attributes or composition makeup of an individual. Its interaction with the environment, or rather the change in expression or change in genetic value due to modulation with the environment, and then the environmental effects per se. In other words, what we can't explain by these two other components. So if we, what our job is essentially to push up this value, this G value, as much as possible. In other words, to maximize the response of a genotype irrespective of the environmental influences. So these are the heritable increases. So as plant breeders, our job is to essentially generate better uh, better the progeny that are better versions than the parentals that the individuals that generated them. And this is a constant cycle, and if you do that for long enough, you will continue to make increases. Most of the literature out there suggests that breeding and genetics account for about in the some of the low end estimates around 20% of the attribution to yield gains. In other words, the genetics alone are responsible for about 20%, and that's a low end estimate and a minority of the estimates. In fact, some of the average estimate is around 50%, right? Here's an example of wheat in Northwest Europe, indicating that, in fact, agronomic management is actually was actually or lack thereof was actually driving our productivity down, and which meant that this is an area where there needs to be significant improvement. And in fact, any yield gains that they have observed vary anywhere from 43% to even 75% in some regions, purely due to genetics. Okay. So plant breeding accounts for about half. That means our job is to increase half of the potential, and agronomists can work with farmers to increase sort of that other half. Okay, and then we can leave some of that up to changes in environment. But how can we then speed up this genetic gain? How do we make sure that we close that yield gap that I've kind of illustrated now? Some of you may be familiar with studies on genetic architecture or genome-wide association studies. They've been fairly common across all of uh, essentially biology or the field of genetics. And what they aim to do is to provide an expanded ability or a more comprehensive ability to identify the causal genes or sets of gene groups that may be responsible for a particular phenotype or trait characteristic in a species. So this is an example of my student Henry Cordova went through the task of essentially collecting a series of data points from this paper, which collected the panel size. These are a series of genome-wide association studies. Each one of these lines represents one for a specific trait as listed there, and then the total markers. What they didn't report were the number of GWAS hits, in other words, the number of genes associated with each one of these findings. So he went and collected each one of these, and this is kind of what we saw. All right. One interesting thing is that you can see here is that our ability to discover new loci, it continues to accelerate. So if we continue increasing the panel size, it seems like we continue to increase. There's some correspondence here with increasing number of genes identified. But um, right, so larger number of the panel size has a big effect on our ability to identify. But there, in some cases, some genetic architectures do result in diminishing returns of markers, meaning that markers are not just the whole story. If we characterize the genome better, that doesn't necessarily mean we'll have better returns and the number of our ability to understand what are the genes underlying them. In other words, it plateaus. And so genetic architecture and complexity at high end sizes, at high panel sizes, does result in some separation. So, in other words, just finding what the genes that are responsible is not the entirety of the story. And this is in the case, and if we move to humans, this was evidenced by this recent study where they identified uh, 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 this group was able to capture enough information to account for most of the heritability responsible for height, which is an additive trait of relatively high levels of heritability. So, in other words, after they conducted 281 genome-wide association studies, 12,011 common DNA variants, they were able to account for over 80% of the heritability for human height. Um, the remainder, this was after 5.54 million individual samples. This is some of the largest genome-wide association studies conducted uh, in all of in any species. And some of these minor variants, which is about 20% of their heritability remaining, are concentrated in some of the lower frequency populations that were undersampled in the study, such as African populations. So what I'm trying to connect here between plants and humans is the scale of sampling required in order to understand at least the majority of the prevalent genetic variation necessary to fully understand the genetics underlying a trait. So this creates a complication for us breeders needing to make progress on a annual basis or on a decadal basis, however you want to look at it, because it becomes quickly obvious that accumulating all of the favorable genes that are associated with a particular trait of complex architecture, for example, such as yield, becomes more and more probabilistically difficult. So we need to implement the best practices that we can in order to arrive at the best possible decisions. What I mean by that is if we we design a system of breeding that we can then implement to collect data and then uh, which will enable us to point sort of in the right direction so that on average, every time we make a selection, we are hitting the mark on some accumulation of these favorable genetics without having to peer very deeply or very much a lot of detail as to what these genes may be. All we know is that we have the right outcome regardless of the environmental influence. So our program works this way. We make crosses. We do crossbreeding. We take parental top elite parents and we make crosses among them. And at this point, we will advance them in the greenhouse, some of the early generations. And this leads us to some Mendelian genetics level traits that are highly heritable, controlled by uh, one or few genes, which we can select for early on. For example, architecture, seed code, and flowering. And then as we advance into further generations, we can move into replicated trials to determine, for example, yield and canning quality, as well as more complex diseases. If we identify a top candidate within at this stage, then we will move into the advanced evaluation stages of the program, where we will mainly focus on the most complex traits such as yield and canning. What in more a little bit more detail, uh, what this means is that if we will start with about 25 to 30,000 for one market class plants at the F2 generation. So I'll go out there in the field and try to identify single plants that are superior in their these attributes, and try to select the top, and then move them on to some of these replicated stages to ascertain their performance as far as yield goes, for example. If this is successful, we will go from 25,000 to maybe one individual that will have accumulated most of these superior genetics in relation to its predecessors. Sometimes we don't find any in some market classes. So you can see we generate all this variation, but we discard 90% plus of it, at least 90%. Yield trials. So this is one. I'm going to focus now on these two complex traits that we focus on: canning quality and yield. Yield trials are conducted usually in small plots. These are small plots of individual uh, genotypes or individuals, and they are conducted in multi-location experiments. They are replicated, and they are multi-year data sets. So by the time we reach a decision to release, this is the case of one of our recent releases, is a black bean called Kona. We will make decisions based on up to 24-year site combinations. Okay, so that at least we'll have high confidence in our decision to say yes, its performance is as tested as widely as we can in relation to some of the standard checks that are available, which are other varieties that we consider sort of that we aim to surpass. And this system works very well because it sets standards. It sets a point that we must meet. So whatever we have to surpass is this is a constantly changing target. The checks that we used 10 years ago are completely different than the checks that we use now, and this process continues um, potentially forever. Now, canning quality is another process. We have a pilot plant here at MSU that we use, and this enables us to evaluate large batches of individual samples. So we will take samples from these advanced yield trials, and then they will go into canning quality trials with using standard sort of canning facility procedures. And we have, with our collaborators here at the USDA, there's established color charts and color and standards for appearance and color for our to conduct these canning evaluations. So annually, we did some this morning actually. We will conduct evaluations for these two traits in the case of black beans. So historically, what has happened is that in the case, for example, here of navy beans, we increase about 76 kilograms per hectare per year uh, on average, while canning quality maintains stable. So now what this represents is not that we can't make progress, say, on canning, but that they are very different traits in and of themselves. Yield is something that we continuously aim to push up, while canning quality is something that we try to tend to maintain at least at some above average level. Uh, this indicates that um, increasing these two traits simultaneously is difficult. So it's better to improve one at a time, and then try to in a following iteration try to improve on that version of that new variety that which has the potential to have better uh, performance in the other traits such as canning. But the other aspect that this represents is that if their our ability to improve, say, canning quality is more limited, that means that the genetics that make up one versus the other are completely different. And again, this poses a challenge. So how do we address our understanding, or developing a better understanding of genetic architecture and background information relating to genetic architecture to develop new tools to conduct breeding. And one way is, of course, through genome-wide association studies. But the way we're uh, sort of innovating in this space is by bringing in um, evaluations or analyses that incorporate multi-trait analysis so that we can understand the interactions between traits so that we can develop tools that represent that interaction, that joint effect rather than individually, as most genome-wide association studies are. So what we did was take a panel of Middle American beans throughout 3 years, and we evaluated them for a number of traits. But in this particular study, we focused on yield as well as other seed seed characteristics and agronomic traits such as flowering, maturity, and lodging. Lodging is when plants lodge lower, such as like kind of like growing along the ground, but because of breakage or some sort of disruption rather than its inherent genetic tendency to grow that way. So we evaluated these traits, and of course, as we might expect, yield was among the lowest heritability, a low heritability trait, um, while some other ones exhibited rather greater heritability, like lodging. So we took this data and we conducted multi-trait analysis, GWAS analysis, and we found some series of interesting interactions where changes in one allelic state of one trait would result in sort of these crossover, either crossover or changes in magnitude effect in another trait. So in this case, days to flowering going up when switching from reference to alternative allele, and then yield going down, and with this in the same SNP position. In this case, there was a change and some interaction in terms of changes in upward trend of both, which is something, something more or less what we would want to see, but in some of the characteristics like yield. So we found a series of these interactions throughout the genome, and this builds a base or a set of tools or a set of information that we can use to build tools for marker assisted selection or even genomic selection for these characteristics simultaneously. Another interesting component um, of this study was our ability to characterize epistatic interactions. So these are gene by gene interactions that also that result in crossover effects, but these are among two different SNP sites. So this SNP interacting with this other SNP, right? When going from reference to alternative allele in the case of days to flowering or yield, right? So when the reference allele is in one position, in one state, switching to other state, and both are in the same condition, there is no change. But when one is in the reference state and the other one and switches to the alternate state, there is an increase. So the interaction causes an increase, but it requires um, it requires a difference in their in the polymorphism, in the SNP polymorphism. So in conclusion, we were able to explain some of the highest a high percentage of variation, up to 52% in some of these single trait and multi-year analyses, including epistasis, and we found variation up to 90.96. So this is just for a few, if you might imagine, how this relates to our traditional way of breeding. If we're increasing to 76% kilograms per hectare per year, this few interactions allow us to push this up just uh, um, by fixing a few of these SNPs by themselves. So that in combination with our average rate of gain, that might put us over that at 2% or so increase per year, perhaps. In multi-trait analysis, some marginal loci, while we identify other more important loci in another chromosome for flowering and yield, and multiple interactions were identified. So this demonstrates the applicability of these approaches. Another study that I would like to focus on is some of the work that we're doing on deep learning and computer vision to better phenotype for canning quality. And this is the work of postdoc Lovepreet Singh in the lab and a data scientist intern, Madhav Lela. Um, so now what we wanted to do here was to bring some more objectivity, more standardization to our ability to rate for a particular for color. This is something that we're trying to extend into other attributes, but we started with color and black beans. So this we know that when we evaluate them visually, this is a process that can be subjective. It could be prone to rater bias, okay? But also conditions, right? So lighting conditions and other factors that are outside the panelist's view, okay? So what we wanted to do is just take a system that we could incorporate to kind of standardize this process. So we have a large bank of images that we've collected over the years for black beans, but also other market classes. And what we did was train an algorithm that would identify beans and segment beans out of an image, which we could then use for analysis just of those pixels alone. So this is just a simple YOLO object detection procedure, and then a segment that which then provides us with a segmentation mask. That segmentation mask was then analyzed across multiple parameters that would be that we would um, uh, calculate Euclidean distances to relative to an ideal reference. So in this case of black beans, we had a pure black reference. The idea was to estimate based on the conditions, the lighting conditions and the conditions at the moment of evaluation, what was the relative distance. So we have here visual rating on X, which is the human visual rating, and then the image based D-score calculation. And in many cases, we can see how D-score resulted in the visual rating resulted in an underestimation of the actual color of beans, whereas in other cases it resulted in a visual rating resulted in an overestimation. So these beans should have moved actually down, while these ones should have moved actually up in the scale. In a minority of cases, they were both were appropriately categorized, but these were obviously a very small number, um, indicating that we had a large proportion in the middle somewhere that needed to be reclassified as either higher or lower, indicating that this was indeed a better procedure to pursue as far as introducing objectivity into the whole process. The other thing that it would able to provide us with was an ability to further discriminate among classes. So humans were rather unable or less capable to distinguish, say, between whole units. If the scale goes from 1 to 5, 5 being the best, humans could only discriminate between, say, a 4 and a 5, and not a 4.5 and a 5, while this system enabled us to further narrow down those confidence intervals at zero point 0.5 scales. So it provided us with an objective, high throughput, low cost measurement of color. It's a simple procedure that can be incorporated in any local computer with simple RGB images, inexpensive as long as the quality of the images is good. This will thus provide more consistency and better selection accuracy, as well as heritability. We went from a 70% or so heritability with visual ratings up to almost 96% heritability with D-score. So this is a significant improvement in our ability to detect a signal versus noise. And it's high throughput. It can image thousands of images in hours. And the idea here is, well, of course, to train deep learning models that can identify also for defects. So before I hand it over to Karen, I'd just like to point out some challenges more and that are kind of relevant to our hosts and to GFI and our group here. In order to drive more driving consumption in North America, particularly, we need to identify new products, new ways of providing options to the consumers, so that they see beans as an alternative to introduce into their diets. So that could be product variety there, but inevitably there needs to be some shift in diets. Um, either from traditional consumers or new consumers that seek to introduce beans into their diets. Regardless of what happens here, in order to meet this demand, plant breeding will play a key role in introducing some of these characteristics, since plant breeding is a highly inexpensive way of deploying a new feature into the into the food system. So, for example, improving color, say in the case of canning, is a way of introducing a new characteristic without the additional need for processing or manipulation. Uh, it also by the same kind of logic or token, finding avenues for breeding for canning quality will be more prevalent. So to produce even cleaner label beans or other products. This is kind of interacts with this issue of complex genetics. So some of the challenges that happen at the agronomics or at the primary production side of things will need better and better tools and better capacity to deal with them if we are to meet an increasing growth in demand. Um, so stacking them is a slow process, but there are there are technologies out there that will enable us to accelerate breeding and develop faster product delivery. From a sustainability point of view, beans are relatively poor nitrogen fixers, and they as petroleum derived products fertilizers are used commonly in high input production systems. So there's an opportunity here also to reduce cost of production and also sort of the sustainability, further increase the sustainability of beans by enabling them rather to produce um, more uh, with with atmospheric sources of nitrogen. I'd like to thank our lab members, our various technicians, postdocs, and students, as well as, of course, Dr. Sihi's lab, and our partners here in Michigan and our federal funders. Thank you very much.
Thank you so much, Valerio. I really appreciate that wonderful talk. Okay, final question for both of you as bean advocates. What is one thing that you want this audience to take away as they consider kind of incorporating beans in their research and their daily lives, all of that?
Eat more beans and become a bean connoisseur. The- if once you start getting into beans, you'll find that you can taste differences among all of these. There's some variation in there as well. Obvious as Karen showed, there's variation in their nutritional properties and their bioavailability, and they have a whole host of different uses. So just it's an interesting sort of area to get into if just as a consumer, let alone as a scientist. Yeah.
Yeah, we'll see if we can get more people in this community to channel beans into meat mimics and things like that so they get out there more. With that, I really want to thank you both for your time today and for this lovely discussion. It's been such a treat, and I really appreciate your time. Um, yeah, thank you so much.
Thank you, Bianca.
Thank you.
So nice to
Bye.
meet you. And for all of you in the audience, we'll see you next month for the next Science of All Protein seminar. Thanks, everyone.